According to new UCLA health research, astrocytes appear to play an important role in obsessive-compulsive disorder-related behaviors. Researchers were surprised by the new clue about the brain mechanisms underlying OCD, a disorder that is still poorly understood. They wanted to know how neurons interact with astrocytes, the star-shaped helper cells that support and protect neurons. Scientists are still trying to figure out how these complex cells play a role in psychiatric and neurodegenerative diseases. Obsessive-compulsive disorder, a lifelong anxiety disorder characterized by repetitive thoughts and actions, affects an estimated 2 to 3% of the U.S. population at some point in their lives, though its prevalence may be higher due to underreporting and underdiagnosis. Although psychotherapy, antidepressant medication, or both are commonly prescribed for obsessive-compulsive disorder, available treatment is ineffective for a significant proportion of patients. UCLA researchers discovered that a protein associated with OCD and repetitive behaviors in neurons was also found in astrocytes by studying the proteins expressed by neurons and astrocytes in mice. The discovery suggests that therapeutic strategies targeting astrocytes and neurons could be beneficial for obsessive-compulsive disorder and other brain disorders. The research has revealed a new cellular mechanism, which not only involves neurons, something that was already known, but also involves astrocytes, working together. Research can now be expanded in this area to cover additional mechanisms and cells. The striatum, a brain region involved in decision-making and motor control, is thought to play a key role in obsessive-compulsive disorder. That is precisely the area of the brain studied by UCLA researchers when they sought to investigate the interactions between astrocytes and neurons. The study's corresponding author Baljit Kak is one of the researchers who has studied astrocytes extensively in recent years, thanks to technological advances that have made it easier to study these complex cells. While previous research compared gene expression between neurons and astrocytes, this new study advanced understanding of the two cell types' interactions by analyzing protein expression. We really have to look at the proteins because they are very complex and diverse. Depending on which cell expresses which proteins, we can predict the functions of that cell, said co-author Jocelyn Soto, a neuroscience PhD student at UCLA's medical school. The researchers used a variety of techniques to isolate and visualize proteins in the striatum's neurons and astrocytes. When they compared proteins found in neurons and astrocytes, they discovered that both contained SAPAP3, a protein associated with obsessive-compulsive disorders. The study's findings were validated by reintroducing the SAPAP3 protein into neurons and astrocytes of mice that had been genetically modified to lack the gene responsible for protein production. When the researchers measured the protein's effects on compulsion and anxiety, two of the typical hallmarks of obsessive-compulsive disorder, they discovered that the two types of cells interacted differently. After the SAPAP3 protein was returned to astrocytes and neurons, the mice no longer groomed themselves compulsively, implying that both types of cells could be viable targets for treatments aimed at reducing compulsion. However, only neurons expressing the SAPAP3 protein were linked to decreased anxiety in mice, implying that astrocytes would not be a good target for anxiety treatments in obsessive-compulsive disorder. Future research will delve deeper into how these cells' interactions affect behavior. More research is needed to understand how astrocytes are formed and maintained. But the unexpected findings of this new study demonstrate the importance of pursuing basic biology questions to help form new ideas about the causes of diseases. The finding that many proteins were preferentially enriched in astrocyte subproteomes has important implications for understanding pathophysiology during neurodegeneration, injury, stroke, trauma and addiction. that are accompanied with altered astrocyte morphology and signaling.